We're going to move on to our next speaker, and thank you to the speakers. We are right on time. This is great. So our next speaker is Dr. Gavin Rumbaugh. He is a professor at the Scripps Research Institute in Florida. And his overarching goal of his lab is to apply knowledge from biological studies of genetic risk factors, such as Syngap, to accelerate development of therapeutic agents to treat impaired brain excitability and behavioral dysfunction in disorders associated with autism spectrum, intellectual disability, epilepsy. We're excited to hear about his work today. Thanks, Gavin.
Hi, can you guys hear me and see my screen okay?
Everything looks great.
Oh, thanks a lot. Thanks for the introduction, Heather. Thanks, Mike and Hans, for invitation. I heard a lot of great things about the first roundtable last year, and so it's a pleasure to be invited to participate with my fellow speakers who I know all very well. So today, I'm going to talk today about our discovery platform for compounds that regulate Syngap protein expression. And I think you got a hint from this from Liz's talk that it's a goal of many of ours because of the nature of the genetic disorder, Syngap1 genetic disorder, that we think finding ways to regulate Syngap protein expression in brain cells will be an important way to treat Syngapians. So today I'm going to talk about a little bit more detail about this platform that we've discovered for unbiased discovery of compounds that regulate Syngap protein in neurons. But before there we go. Sorry about that. But before I do that, I just want to spend a few minutes on kind of the way that we conceptualize Syngap1 disorders. I'm a basic scientist. I'm a neurobiologist, neurophysiologist. And my general training and interest is in understanding how genes regulate functional neural circuitry to carry out important neurodomains that we all cherish, such as in memory and planning and language and motor movements and sensory perception, and how these processes may go awry to cause problems that we see in the kids, including seizure. And so, generally what my lab is interested in understanding is how does Syngap1 gene carry out critical neurobiological functions to lead to a normally functioning brain. And so the converse of that is how do you, when you lose one copy of Syngap1, what are the pathological consequences to the brain, or what are the functional consequences to the brain that leads to altered memory, planning, language, and so on. So that's generally how we conceptualize the disorder. And so you work from many labs over more than 20 years now. Has really identified, Mary talked about 3 functions for Syngap within the Syngap in the synapse from a biochemical perspective. But we see 2 general functions of Syngap1 from a cellular perspective, and how these functions of Syngap1 may help to organize functional circuitry that carry out important neural domains. So as you heard from Mary, so Syngap is an important constituent of the postsynaptic density, principally in dendritic spines and in excitatory neurons in the forebrain. And so there it regulates a multitude of functions to regulate synaptic strength, and we think to normalize both development of neural circuits, neural circuit excitability, but also neural circuit plasticity. And so another function of Syngap that we've principally articulated to the field over now in five different papers is when we look in various areas of the brain, we always see changes in dendritic architecture. And so that's important. And we don't think that these changes in dendritic architecture are related to alterations in Syngap function, to synaptic function. And the reason is, depending on where we look, we may see, for instance, in upper laminas cortical sensory neurons, we see that in Syngap1 heterozygous knockout mice, which model for the genetic disorder, we see or what are called arrested, so neurons that underwent arrested development. So these are much smaller neurons, and they never make it to the right size, so they have fewer connectivity onto them. But just a few hundred microns away and deeper lamina of the same part of the cortex, you see actually in developing animals, you see much more mature, much larger dendritic arbors in Syngap1 mutant mice. And so, the point is that there's a function of Syngap, and we don't understand the molecular functions that lead to the changes in dendritic architecture. But the point is that these changes in dendritic architecture affect the way that functional circuits develop. And you can imagine that alterations in synaptic function and plasticity are superimposed onto a background of altered connectivity of neural circuits. And we think that it's the combination of these two substrates in the developing brain that leads ultimately to the changes in behavior and adaptive functioning that defines this SYNGAP1 disorder. And I would mention that we've also seen alterations, pretty significant alterations in dendritic arborization and architecture in human neurons derived from iPSCs that we published just a few months ago. Okay, so the way that we generally conceptualize SYNGAP1 disorders is it's a developmental encephalopathy where there's altered brain function, and principally altered brain function is going to affect different domains that are responsible for everything that we do, which would include cognition, valence, whether something is good for us or bad for us, of course, social processes and arousal and regulatory functions that are important for learning. And so the way that most genetic neurodevelopmental disorders are conceptualized is that what happens when you lose or when you change the function of these genes, that you alter this functional neural circuitry or functional networks. And these changes in functional networks are what underlie the symptoms that we see. And the way that I conceptualize SYNGAP1 is that epilepsy is an outcome of this altered functional neural circuitry. So just as altered cognitive function is an outcome of this, I see the epilepsy also is an outcome of this generalized disruption to functional architecture. And so we think principally based on prior studies that SYNGAP1 is particularly important in projection neurons from the cortex, so glutamatergic neurons, the spiny neurons, excitatory neurons. And so we're trying to better understand how to conceptualize this. And just a flavor of what the lab is working on now from the biological perspective is that we're trying to understand how SYNGAP1 affects functional circuits that are important for how we understand the world and how we make decisions. And we want to do that because after talking to many parents, we realize that there are some altered behaviors and also pretty significant alterations in sensory functions. If you think about sensory function, pretty much everything that we do is guided by how our brains map the external world. And so if the brains of Syngapians are unable to process the sensory function, you can imagine that they would have problems learning or defending themselves or basically keeping themselves safe. So the way that we're doing this is to take SYNGAP1 animals and record from various areas of the brain as they do what are called ethological tasks, or tasks that they would normally do in the wild to help survival. And so I think some of you have seen some of this recently in other meetings. And so hopefully when I talk to you again in a year or two, we'll be able to show you a more holistic view of how we think SYNGAP1 is affecting distributed brain function. But today, I'm going to talk to you principally about this platform, about how we're looking for small molecules or drug candidates that can be used to raise SYNGAP protein expression. And why do we want to do this is one of the attractive features of SYNGAP1 from a therapeutic perspective is that nearly all of the patients have very clear loss of function variants, meaning that they have genetic haploinsufficiency where essentially one copy of SYNGAP doesn't work as well as it should. And what you end up with is less protein in neurons in the developing basically throughout life. And so having that reduced protein is what ultimately is the root cause of SYNGAP1 disorders. Even for patients that don't have clear loss of function variants, those that have very clear clinical phenotypes, some preliminary studies we've done in the lab with their stem cells suggests that they also indeed express less functional SYNGAP. So we really do view this as a disease of too little SYNGAP protein expression. So therefore, it would be very, very important to find therapeutic approaches be targeting at trying to raise SYNGAP protein levels. And that's one of the reasons why I'm so excited for people, new investigators that come into the field like Liz Heller, who has really, whole career is try to understand how you regulate gene and protein expression. So we need expertise like Liz to be able to help solve this, this problem of how to raise SYNGAP protein expression. So as Liz mentioned, we know very little. In fact, we know almost nothing about how SYNGAP is regulated. And so, basically we decided to take a step back and say, okay, what might be the ways that we might be able to do this? So we just conceptualize it as very simply is that this is a disease of low protein expression, and that if you have this low protein expression leads to disease phenotypes or altered clinical manifestations. And so if we restore protein levels, we think that we would be able to improve outcomes. And of course, we published many papers in animal models doing genetic gene therapy and showing that we can recover important functions of Syngap. So currently there are various ways that this can be done. And so what we're working on is what we call Plan B, because it's an alternative in a parallel track to Plan A, which is being undertaken by various academic labs around the world, as well as several biopharma companies, which is to use ASOs or antisense oligonucleotides to trick Syngap into expressing more of its protein. It can do this through skipping poison exons or targeting a natural antisense transcript that might suppress expression of Syngap. And these are very exciting technologies, and I hope that they work, but there's no guarantee that they're going to work well. And even if they work, they might not work for all patients. And so it's important to have other ways to do this. And so what we plan to do is to just work on understanding, sort of in the same sense as what Liz is trying to do, except we're going to take an unbiased approach to find perhaps any pathway, any druggable pathway that can ultimately lead to increased Syngap steady state protein. And so there's many different potential druggable pathways that could lead ultimately to more Syngap protein, steady state protein in the cell. So you can, for instance, you can regulate the amount of mRNA that's made. You can alter how that mRNA is regulated, so more gets loaded into the ribosome to make more protein. And importantly, there are all types of regulatory pathways within the level of initiating translation of mRNA. And of course, we know in neurons that protein degradation is incredibly important for regulating protein levels. So there are several documented potential druggable pathways at the level of protein degradation. So if you could increase or boost these pathways, you might get more Syngap protein. Or for example, if you could find a pathway to degrade Syngap, if you inhibited that pathway, you might end up with more Syngap protein. But since we don't know anything, we decided to take an unbiased approach. And this is that if we just created the right kind of assay or the right type of system that tells us how much Syngap is in neurons, if we put enough biologically active small molecules on neurons, we might be able to find that one important compound that indeed could raise Syngap expression. So the ingredients for a successful Syngap boosting compound program are basically there are three distinct elements. One is you need a great assay. So you need a great system that is disease modeling and can tell you how much Syngap is inside of brain cells. You need a specialized facility with robotics that specializes in what's called high throughput screening, where you can take this great assay and then use individual small molecule compounds, tens of thousands of them, against this assay to find those ones that do what you want, which is in this case to raise Syngap protein. And then importantly, you need an ecosystem or a research environment where you can take a compound in your assay that may raise Syngap protein. Then you need to validate it, biologically validate it, to show that it actually raises it in a context that's meaningful and that can actually change and improve disease-related phenotypes in various models, whether they be rodent or human. Okay, so the system we've been working on now for almost 9 years, and the reason why is because we had to reverse engineer in some ways high throughput screening, because we have to do this in neurons. And why do we have to do high throughput screening in neurons? Is because Syngap, while the message may be present in many tissues, and I know that I get a lot of questions about this, there's very few documented examples of protein existing outside the brain, and even within outside of neurons within the brain. So even astrocytes within the brain, we see very little Syngap expression, tons of message, very little Syngap protein expression. So in our experience, if you wanted to understand the regulation of real Syngap, then you would need to measure expression from neurons. And in particular, you'd want to do this in neurons that only have one good functioning copy, because it's very likely that when you lose one copy of Syngap1, the cell undergoes a state change where it's now coping and compensating for having only one functional Syngap copy. And it's entirely reasonable that the druggable pathways that exist in a cell with only one good copy of Syngap may be different from the druggable pathways in a normal cell that expresses 100% of Syngap. So you would need to do this in neurons that lack one good copy, and you would need to be able to track the Syngap protein in real time that's made from that one remaining good allele. So what we did was we came up with a system, what's called a dual luciferase system, where we make a mouse that expresses one type of luciferase. This just tells you how many neurons are in your assay plate. And then we paired that with a specific knock-in mouse where we knocked a luciferase reporter tag with only 11 amino acid tag into an internal exon within the SYNGAP1 mouse locus. And what you end up with is a tiny 11 amino acid insertion inside of endogenous SYNGAP protein, and then that can reactivate a split luciferase reporter, and then the amount of luciferase signal is directly proportional, in theory, to how much endogenous SYNGAP is made in those neurons. And so this is all on top of this mouse here. This background luciferase mouse is crossed onto a homozygous background of a SYNGAP1 conditional allele. So we can we can destroy one copy of SYNGAP1 in this culture system by just adding a little bit of AAV virus that expresses CRE to induce haploinsufficiency. So this system checks all the boxes for a good disease modeling assay. And here is just some data showing that the luciferase reporter does a very good job at tracking in a very, very easy and straightforward way and a very highly quantitative way the amount of endogenous SYNGAP that's made. So this is roughly where we inserted the luciferase reporter. This shows you that the insertion of this reporter doesn't change endogenous SYNGAP expression. But here you see in the heterozygous knock-in mice, this tag also includes not only a luciferase tag, but a flag tag. And you can see that you only see signal in the heterozygous knock-in mice that have the tag, but importantly, it's the correct length, 135 kilodalton. And you can also show that we can use the luciferase reporter on immunoblot to show that it's also present only in these knock-in mice. This is a quantification of SYNGAP protein, total SYNGAP protein from these mice. Importantly, we don't see a change in endogenous SYNGAP expression, so the tag appears benign, at least in terms of SYNGAP expression. And then we did correlations between how much protein we add and exactly how much signal we get, both with the SYNGAP antibody as well as this N-bit signal, which is a luciferase reporter. And you see that the reporter does as good of a job as SYNGAP antibodies to report the relative amount of SYNGAP protein that's present. So we're pretty excited about this mouse. And then we had to take it and adapt it to high throughput screening assay plates. And so what we do is we take assay plates that have primary cultured neurons in them, and each assay plate has 384 wells. And so what you could then do is to then use a high throughput, a very homogeneous luciferase assay that takes just a couple of steps in 10 minutes. And what you end up with is you get in these assay plates, you get 15 to 20,000 units of fluorescence, sometimes even more, which results in an incredibly high signal to noise for SYNGAP protein expression over background, and gives us metrics that are indeed more than what we need to do high throughput. Importantly, in this assay format, we wanted to know if it can tell us relatively how much SYNGAP is there with relative precision. And so what we did is we plated either heterozygous or homozygous luciferase reporter knock-in mice. And we would expect if this worked well, you'd get about twice as much signal in the homozygous mice because they have 2 copies of the tag SYNGAP relative to 1. And in fact, that's exactly what we see. We see, essentially a doubling in the luciferase signal, indicating that we think we have a pretty good system for tracking in a very easy and straightforward way the amount of relative SYNGAP expression. So we have a good assay. And we can do this in primary neurons, but the problem is that you need a facility, and you need to convince this facility to take your system and adapt it to high throughput. Luckily, Scripps has one of the most well-respected academic high throughput screening centers. They've done more than 300 full deck million compound micro HTS screens across more than 100 NIH grants and over 100 papers. There have been 77 identified molecular probes. So that's meaning that new compounds that can modify a particular protein to regulate cell biology. And importantly, Scripps has more than a dozen FDA approved medicines, most recently tafamidis and ozanimod. And ozanimod is interesting, is that it actually came from the libraries, one of the libraries in the Scripps High Throughput Screening Center that I'm using, and we will indeed be using a lot of the same libraries to look to see if they can regulate SYNGAP expression. So the important thing is we have a facility that has a track record for doing high throughput screening and identifying drug candidates. And so what I had to do is convince the team in the screening center was to take this mouse-based system we have and to adapt it to their high throughput industrial robotics. And so we published a paper a few years ago showing that we, in collaboration with Tim Spicer and Louis Scampavia, who are the directors of the Scripps Screening Center, that indeed were able to take this mouse-based primary neuron system and scale it up to assay plates that are compatible with high throughput screening. So here we use genetic knock-in mice that express synaptic reporters, because this allowed us to not only test the ability to if we can do it, but how well can we track the development of a metric of a healthy neuronal culture. And so synapses are one way to do that. And we see very low variance between wells in an assay plate. That made us very happy. And the synapses are much harder to measure than say luciferase, which is a homogeneous signal from each well. So we were very excited about this. So we were able to plate neurons in such a way that can be used on high throughput robotics. But can we get enough throughput in order to screen the 100,000 compounds that we think we would need to screen in order to find robust Syngap regulators? And that can be easily overcome by a reasonable sized mouse breeding colony where we get just 2 to 4 litters per week. And so within those, if you use EVA 3D4 or even a 1536 well plate, we're talking very easily, just a few hours of work a week, you can do thousands and thousands of assays a week. So over the course of a few dozen weeks, you can get to that 100 or 200,000 compounds relatively in a relatively painless way. So what we did is once we convinced the facility to help to collaborate with us to convert this to high throughput screening, we did a pilot screen. So you want to know, so yes, your assay may work by showing you that there's more or less Syngap expression in there when you do heterozygous or homozygous knock-in mice, but how does it perform when you do an actual library? So in order to test it's more of a dress rehearsal for how well does my assay work in high throughput screening, we used 1,000 compound scout library. And this library is interesting because it's made up of very small molecules that was designed by a colleague of mine, Chris Parker, where all of the constituents of this library are known to bind within binding pockets for druggable pathways. And so some of these compounds, many of these compounds have also a diazarine moiety, and diazarines are interesting because they can, using UV light, you can covalently couple the compound to the protein it binds to, and it makes it much easier to figure out the mechanism for how you may be changing the biology that you're interested in this case, changing SYNGAP1 levels. And so what we did is we did this very easily, just in 1 or 2 weeks. And we did each assay, each compound, 1,000 compounds, each compound was done in quadruplicate to understand how reproducible the data are in this format. So what did we find? So remember, there's a background luciferase, and this background luciferase tells you how many neurons are present. And that's important because we want to know, if these compounds are toxic or just generally regulate Syngap expression. So for instance, anything that dramatically increases or decreases Syngap expression in this background firefly luciferase, would be less interesting to us because it might just be doing something wonky to the cells. So in every assay plate, we have DMSO controls, which are in this band. And this gray shaded region here is what we call the hit index. So anything that falls outside of this gray shaded region is significantly regulating the reporter, in this case, firefly luciferase. So in DMSO control, we wouldn't expect any outside this band, and we don't do that, which tells us that we don't get any false positive, which is great. All these dots are the 1,000 compounds. There's 1,000 dots here, which represents each unique compound. And you see that indeed you have some increase and some decrease in the background luciferase signal, which is not expected. But for every well, we have 2 signals. We have the background luciferase, but that well also contains a signal for Syngap, which is this N-bit signal. So what we're able to do then is to try to compare the results across both data points to find those compounds that regulate the Syngap signal but don't do anything to the firefly signal. This inset here is important as it shows that these were each data point you see here is 4 individual assay wells for the same compound. There are no error bars because essentially there's absolutely no variance. So this is data from plate 1 versus data from plate 2, and you see almost a straight line, meaning that we get essentially the same data, we get the same signal across every plate. So that tells us that we really only need to screen in 1 or 2 wells when we do the high throughput screen. Now, what did we find in this pilot screen? Because it's possible in the first 1,000 compounds that we may find something interesting, and indeed we found several interesting things. So because of the ability to plot the Syngap signal for each data point versus the background signal, what we're looking for are compounds that basically up or down regulate the Syngap signal with having no effect on the luciferase signal. And indeed, and this would be present in this band here, right? So if you see a compound way up here, that would be a 50% increase in the Syngap signal with no change in the luciferase. Unfortunately, it's always much harder to find upregulators of any biological pathway. We found a couple that were borderline hits, But because this was a pilot screen and we were really testing the chops of our assay and our system, we focused on these what looked like selective downregulators. And so downregulators are interesting in the sense that they could be inhibiting pathways. They could be regulating pathways that can depress Syngap, but if you are able to manipulate those pathways in the opposite direction, you may be able to then take that pathway and then trick it into making more Syngap. And so that's why we think it's important to not only find these compounds and then figure out how they work, what the mechanisms are, because the whole point of this is to elucidate potential druggable pathways that can be used to change Syngap expression. Okay, so we found 7 or 8 potential downregulators, and so that we then did a follow-up dose response study. So this is the typical drug discovery workflow: is that you take your hit from your high throughput assay, you get fresh compound, you dissolve it, and then you do a concentration response curve. And interestingly, what we found is that in this dose range here, where we see no effect in the background luciferase, we again repeat our data where we see a dose-dependent increase, or I'm sorry, a dose-dependent increase in Syngap inhibition. And we see this of 5 of the 7 compounds, which was pretty exciting to us, indicating that our screen is pretty good at pulling out compounds that actually move the reporter in a particular direction, and it's reproducible. So we're not going to get a lot of false noisy data points from our high throughput screen.
And 5 minutes, Gavin.
Thank you. I'm almost done. Appreciate it.
Perfect.
Yeah. And so we were particularly excited by this is because I mentioned that some compounds were plus and minus the diazarine covalent modification motif. And in fact, this 219154 compound is indeed the compound plus and minus diazarine, and the library was designed in such a way that the diazarines should not interfere with binding pockets. So that told us that we pulled out both of these. So this is a particularly interesting compound to us. So we're pretty excited about our assay. We were just getting ready to do screening at high throughput for all of 2020, but of course COVID got in the way of that. So we are just now getting back up to speed. And so to give you an idea of how the screen is going to work, it's going to encompass pretty much all of 2021, where we do 1 liter per week per staff member, and we have 2 current staff. And so if we do each compound in duplicate, as I mentioned, if you do the math for these 384 well plates, you can come up with, after 40 weeks of iterative screening, of 102,000 compounds. And so that's important because 102,000 compounds, based on prior data from different types of high throughput screens, should yield several dozen validated Syngap regulators to choose from that may be most efficacious. And then the last slide I'll leave you with is that then it's one thing to be able to find these compounds, but as I mentioned, you have to have a system and an environment to be able to validate them. And so most of what my lab is known for is developing models to understand how Syngap haploinsufficiency affects neural structure and function. And so we can use that knowledge as ways to validate as a platform for validating the compounds that we pull out. So for instance, we now have human iPSC models. So if we found a compound that raises Syngap protein in mouse neurons, so does that also raise Syngap protein in human neurons? Does it raise it in human neurons that come from iPSCs from patients, for instance? And then what we would want to do is take those compounds and try to reverse various cellular and network measures that we know from our mouse models results from Syngap haploinsufficiency, such as changes in synapse function, memory, and behavior. And so with that, I would like to just thank, for this project in particular, the co-directors of the Screening Libraries Center at Scripps, Florida, NIH, who's been funding this now for 3 cycles, and as well as other funders. And in particular, I'd like to thank Syngap Research Fund for funding a postdoc in the lab. While they're not directly involved in this project, they are going to be involved certainly in studies to validate the compounds that we pull out from the primary screen. Thank you very much.
Thanks, Gavin. That was wonderful. That is really exciting work. And we're excited for 2021 to see what comes out of this. There are a couple of questions in the Q&A. We have time for, I think, 1 or 2. I think you answered Janie's question during your talk. She says I typed that too early. So Janie, please update if part of it wasn't answered. And there's a question about whether the compounds that you're testing in this library are known to cross the blood-brain barrier. I'm guessing those aren't, but do you have an idea how many are?
Yeah, so there's a million compounds to choose from, and that's not an exaggeration. There's literally, more than a million compounds. And so one of the jobs of the center is to match the sub libraries to the project you want. And so there are some, on the face of it, you would say, well, it's a brain disorder. You want to only screen compounds that have, blood-brain barrier penetrance. However, we also work with medicinal chemists. And they tell me that that's not a big barrier, that if you have an active compound, a backbone that does not cause blood-brain barrier, you can do downstream derivatization to help them cross. But I should also mention that's one of the reasons why you want to screen so many compounds is you need to have what are called different chemical structures or backbones to work with, right? So some of them may be really great in cells to regulating Syngap expression, but no matter what you do, you can't get it to cross BBB. I mean, that's a dead candidate. So then you need to go to candidate 2, candidate 3, candidate 4. So.
Right. Well, it's great that you can screen so many. That's a really nice high throughput system. And then one last question. In addition to finding drugs that modulate the disease, can you use your system to validate ASOs or other similar treatments?
Yeah, that's a good question. So, I've given this talk a few times and I always forget. So I forgot to mention, and also this is relevant to SRF. So one of the things our SRF fellow is doing is to, we've created the same Luciferase screening platform. We made a variant, we made an insertion into an iPSC line that comes from a patient, and we did that allele specific. So we put the reporter tag into the non-mutated Syngap allele, and it reports Syngap protein really nicely in developing human neurons. And so while this platform, while my screen per se won't be good for that, we're developing tools to support the platform that can be generally useful for the community to be able to track Syngap protein. So if somebody was interested in ASOs, they could have our iPSC patient cell line, and they would have a very easy way of tracking endogenous Syngap protein expression.
That's great. Thank you. Perfect. I think the questions were answered, and we should probably move on to our next speaker. So